I'm Beth. I'm a project scientist at the UC Davis Genome Center, and I work in the lab of Dr. Richard Mitchell-Moore. So I'm really grateful to Rosalie and to Milo Mitchell for giving me the opportunity to participate in this series. One of my favorite things to do is to talk to young people about science. And I thought I'd start by telling you a little bit about what a project scientist is, because it may not be exactly clear from the title. And a project scientist is somebody who works in an academic research lab, somebody who has a PhD and several years of experience and is therefore able to conduct independent research, although project scientists usually participate in their own research projects and then also in collaborative research projects with other members of the lab. The lab is usually headed by a university professor, and a project scientist is working with graduate students, so master's, PhD students, and bachelor's students, and technicians, and to conduct research in the lab. And not all universities have project scientist positions, or if they do, they might go by other names. But that's the basic idea of what a project scientist does. So a project scientist is also capable of designing entirely new research projects and seeking independent funding for those. So I work in the lab of a professor here at UC Davis who, as I mentioned, Richard Mitchell-Moore. We are a lab that studies lettuce. And you might ask, why lettuce? And lettuce is actually more exciting than you might think, because it's something that is one of the most consumed vegetables by Americans. And this is not only because, so you might go into a store and buy it and take it home to make salads, but if you go out to eat, if you go to your favorite fast food restaurant and get a burger, they usually put lettuce on top of the burger. If you go to a deli and get a sandwich, they put lettuce on the sandwich. So you end up consuming lettuce in a lot of different ways, and it ends up being a pretty important component of the diet. And this is really important because Americans as a whole do not get their recommended intake of fruits and vegetables. And so lettuce actually represents a major vegetable and therefore a major source of nutrition in the American diet. So that is one of our main motivations for studying lettuce. And it's interesting for a number of different reasons. And one of the things that we are doing as a whole lab group is we are interested in studying which genes are responsible for which different traits that might be useful for agriculture. So we are interested in understanding these as a fundamental process of biology, but in the process we are generating knowledge that will be useful for other researchers, either in academia or in commercial agriculture. So I brought some different kinds of lettuce to show you. So I mentioned we're interested in studying genes that are responsible for different traits. So nutrition is one trait that I mentioned is really important. So we have a student who is working on improving nutrition in lettuce. We have a lot of people who are interested in improving disease resistance. There are many different challenges that will come about with climate change, and we need to make sure that we can keep continuing to grow and produce high quality lettuce for people to eat in the future. And so that is kind of what we're doing as a lab group as a whole. And I have my own specific research questions that I'm interested in. I wrote them here on the board. I don't know how easy it is for you guys to see it, but I will say it. So one thing I'm interested in is how you get different combinations of traits. And the second main question that I've gotten into recently is, how is it that you can give seeds long lives? And both of these questions are very important, and it's not as if there's just one answer to the question. So If there was, it would be pretty easy because then we could say, okay, we found the answer, let's go on. But really the answer to each of these questions, I like to consider you to think of it as a kind of a puzzle. So I like putting together puzzles. They're fun jigsaw puzzles at home. But when you have a jigsaw puzzle at home, you have all of the pieces and they're already pre-cut into ways to fit them together. And you know what the picture is and you're putting them together. So here we have a general idea of the picture. We don't know how many pieces there are, and we don't even know what the shapes of those pieces are and how they might fit together with other pieces. And so because each of these pieces is actually quite complicated to figure out, so you as an individual scientist may spend a number of years or even an entire career just working on figuring out one piece, the shape of this one piece of the puzzle, and then we come together as a community to figure out all the pieces that we have and see how they fit together. And then at some point, maybe we will have found all of the pieces, and maybe we will just find that there are still more pieces that we've been missing. So, so I will talk a little bit about the pieces to the answers to these questions that I'm working on. So the first question about how we get different combinations of traits and why that's important. So I already mentioned that we are interested in studying traits for a number of different reasons. And we have a bunch of different traits. So this is a red leaf lettuce that I'm holding. I have a green leaf lettuce. Sorry, my camera's reversed so you can see the writing, and it makes it hard for me to figure out how to put things into the camera. So this is green leaf lettuce, and then I have a romaine type lettuce and a butterhead. I don't have a crisp head type lettuce like iceberg to show you, but I think everybody kind of has a good idea of what iceberg lettuce looks like. So these are all different cultivar types of lettuce. So even within these types, there are many different varieties, but they're all the same species. They're all Lactuca sativa, cultivated lettuce. But if we want to find a new source of disease resistance or tolerance to drought, a lot of times we don't already have those traits in our cultivated types, but they do exist in the wild relatives of lettuce. And so I wonder what you guys might think a wild lettuce would look like. So, so you're very familiar with the cultivated lettuce that I've shown you. So I just want you to think about what you might picture a wild lettuce to look like, and then I will show you what it looks like. Okay, do you have your picture? So I'm going to show it to you. Ta-da! This is wild lettuce. So, so it's really big. I'm going to move it back so you can actually see how tall it is. So this plant is actually taller than I am. And if I move it closer to you, you can see that, so the leaves are kind of tough. They also have like these little spikes on them. So if you came across this plant in a field, like, I don't know if you would look at it and think this is something that I want to eat. So, so if we want to take advantage of the traits that exist in the wild lettuce and introduce them into our cultivated lettuce, we want to make sure that we can get the combinations of traits that we want, and combinations of traits that we don't want can be avoided. So, so if you had a trait for tolerance to heat stress, and or if you had a trait that improved the nutrition, but it also came with the spikes, you know, that would not be very good for you to eat. So there are many different things that affect how easy it is to get different combinations of traits together. And one of them is whether they're on the same chromosome or not. So you guys may have learned about Mendel and independent assortment. So if you have genes for traits that are on different chromosomes, it's much easier to get them to occur in different combinations. But if you have genes for traits that are on the same chromosome, then it's a lot harder to separate them. And so And that is actually the process that I am interested in studying. And it's a molecular process. It happens during, so one way to separate traits that occur on the same chromosome is a molecular process that happens during meiosis. So you guys may have learned about meiosis and mitosis. So meiosis is a specialized round of cell division that will end up giving rise to the gametes. In the case of plants, that's going to be pollen and ovules, and those pollen and ovules come together during fertilization. That makes a seed, and that gives you the next generation of plants. And during meiosis, you also have the opportunity for a genetic exchange. So, so you might remember that chromosomes come in pairs. So if you, so for humans, for example, you have 23 different pairs of chromosomes. And so, and the pairs are the chromosomes that are the same chromosome, but you have the version that the chromosome that you inherited from your mother and the one that you inherited from your father. And the same thing happens with your mother and father plants. And if, and so if you have two different traits, one coming from the mother's chromosome, one coming from the father's chromosome, and you want to be able to, for them to be on the same chromosome, what has to happen is you have to break these chromosomes apart. And it is pretty fascinating that this already happens during meiosis naturally. You have the chromosomes being broken and put back together. So then you end up with a chromosome that now has a portion of the mother's chromosome and the father's chromosome that gets passed down to the progeny. And so my research actually studies the molecular process that is involved in this exchange. So that's for the first part of, that's sort of the first question. And the second question about what gives seeds long lives. So this is also a very important question because you want to, once you have seeds that produce a type of lettuce that you like, and you want to then keep storing them and keep growing them over multiple years. Or if you have seeds from the wild relatives and you don't grow the wild relatives all the time, you want to be able to go back to those seeds and grow them if you need to, and they need to be able to germinate. And some species have seeds that can retain the potential to germinate after many years of storage, sometimes tens or hundreds of years. So I think I shared an article that in some cases, many thousands of years, seeds can retain the potential to germinate. But for most commercial species, for crops that you might eat, it's usually limited to a few years or maybe 10 years. And what we do know about it is, of course, how you store the seeds is going to be important. So so we know that keeping the seeds cold and dry is a good way to retain their potential to germinate. And for this reason, we have seed vaults all around the world where you can store seeds for use in the future so that we don't lose whatever genetic variation and information is contained in those seeds. But I'm really interested in biologically what's happening and why it is that some seeds can retain the ability to germinate even after long periods of time, and others don't. And so that those are my two main questions. And so I think what I'm going to do now is I'm going to talk about, so these are things that I've been doing as a project scientist, but I'm going to talk about how it was that I got to be in this position in the first place. So I am going to share my screen. So I have a few slides to go through. Is everybody, is it clean? Okay. So I'm going to go back and tell you a little bit about my life. So I grew up in Carmel, California. So this is where it is. And this is what it looks like. It's a city by the beach. And when I was growing up and when I was in high school, I was always really interested in nature and I liked science and I did pretty well at it, but I never really thought of myself as being a scientist. I always thought that that a scientist was somebody who was smarter than me or just somehow different from me. I just didn't really have a picture that being a scientist was something that I could do, and even though I really liked learning about nature and learning about biology. So, so what I wanted to do as I was finishing up high school and going to college was I wanted to be a journalist. I really liked writing. I liked that investigative component. So I guess that kind of does carry over into science as well. And I applied to a lot of different universities to go and study journalism. I applied mostly to smaller schools, liberal arts type colleges and universities where I could get an education in writing and journalism. So I ended up going to the University of San Diego. So you guys might know, if you're familiar more with the UC San Diego or the San Diego State. So this is a much smaller university and focused on liberal arts. And but it was actually kind of interesting. So my parents had moved out of the state right before I was going to college. And then the way that this university works is that everybody who comes in as a freshman has to already decide on what major they want. And they get put into a special class with other students who are intending to go for the same major. And you are supposed to fill out some forms to indicate what major you want to study before going there. But they mailed these forms to my parents, and somehow in the move and everything, they were lost. And I never even knew that they existed. So when I arrived to start university, I had not registered for any classes. I hadn't chosen a major. Everyone else already had. And then I had to visit the registrar and the dean's office to get it all sorted out. And unfortunately, at least at the time, it was very disappointing to me that all of the journalism and communication classes were all full. They said you can't actually start with this as your intended major because they're already full. So then they were looking at my transcripts and they said, okay, well, you did pretty well in biology, so maybe, and we have space in biology. So do you want to major in biology? And I was kind of like, well, I guess. And I always thought like, okay, well, maybe, I could, because, they do give you the opportunity to switch later. So maybe when things settled down in the next semester, if I wanted to move over into communications and journalism, I could do that. And so that was kind of my plan when I started after being put in this position where I was kind of forced into biology. But luckily, with that first class, I had a really great teacher. His name was Richard Gonzalez. And he, in this class, really got me to think about biology in a different way than I had before. And I realized that it was something that I was really curious about, and that it was something that was going to take a lot of hard work to learn more about, but that it was really worth it, and that it would be worthwhile to continue to major in biology, even though I really had no idea what that would mean for a career later, or how I could, what I could do with that. I just knew that this was something that I really enjoyed and I wanted to know more about. And so I stuck with it. And then by the time I was getting ready to graduate, I was really interested, still very interested in doing research. I loved all the lab classes that I got to do. And, but I didn't really know what I wanted to do. And I thought maybe, maybe I could continue on and go to graduate school. So I took all of the exams and kind of had that as a possibility, even though I wasn't really sure that that's what I wanted. And what I ended up doing is after graduation, I got a job in the biotech industry. And this was because I had, looked around and, they seemed like good jobs with really interesting possibilities. And the job I ended up getting was at a place that was making test kits for diagnosing autoimmune diseases. And I got to really learn a lot while I was working there. But one of the other things that I realized while I was working there was that even though I enjoyed learning new things and participating in the company, what I really wanted to do was to be able to design my own experiments and conduct my own questions, because a lot of times I would have a question that would be like, well, why does this happen this way and not that way, during the course of what I was doing for the company. But if they were not questions that were really central to what the company was doing, then I didn't really have the opportunity to do that. But then I realized, well, if I'm doing academic research, then that might be a possibility. You give me the possibility to investigate, all these other types of questions that I have. So, so since I, had already kind of prepared for that possibility, that year I decided to just really focus on it and start sending applications. And I applied to a number of different schools, and I ended up getting accepted into a PhD program at the University of Washington. And something that I was not aware of until I was getting ready for applying to graduate school was that, unlike other post-bachelor's education, these programs usually do not require you to pay a lot of money, so you can actually get paid to while you're earning your degree, your master's or your PhD. And this can, so you do have to pay some tuition, but you are often supported through either through fellowships or through teaching assistantships, or if you're working, if you're studying in the lab of a professor that has funding to support students, then you can be supported in that way. So you can actually, it's not a lot of money, I will say, but it is enough so that you don't have to have a separate job and you don't end up owing a lot of money. And I think that this is something that's really important because we really want to have as many people participating in science as possible. And certainly when I was growing up, I thought that all of these higher education programs were things that were going to be expensive and that you had to pay for, and I wasn't sure how I was going to be able to do that. And if I had stopped there and I hadn't actually obtained this information, I probably would have never applied to graduate school. So I'm hoping that that finding different ways to get this information out and will hopefully encourage people to apply for these types of programs if they think that that's something that they want to do and not have to worry about those concerns. So about my PhD, so I really wanted to study cell organelles. So you guys know cells, the basic building blocks of life. They have smaller compartments that do different jobs within the cell. And and some of those compartments actually have their own DNA that's separate from the DNA in the nucleus. So like the DNA that's responsible for making up you or a plant, or most of the trace that you see is usually in the nucleus, but there are other organelles that actually have their own DNA that have a limited number of genes. So the chloroplasts that are responsible for photosynthesis, they have their own genome, their own DNA, and then also the mitochondria, which are responsible for producing energy. And at that time, I didn't really necessarily want to study plants, but I was really interested in these organelles. So I ended up studying with Dr. Arnold Bendich, and he was interested in knowing more about the processes that maintain the DNA in the cell organelles. And because it was a lab that was studying plants, and I started studying plants, I ended up then really wanting to know more about them. So I got interested in volunteering in the greenhouse. I took a number of other different classes, and that was really kind of that was where I got hooked on plants, because I realized that plants do things in a lot of different amazing ways. They have a lot of different challenges compared to animals, and I think it's really fascinating. So I've been hooked on plants ever since. So that was my PhD, and then while I was finishing up my PhD, I wasn't really, again, sure what I wanted to do, and there are a number of different things that you can do with a PhD. You can go into industry, and there are a number of different types of jobs that you could do with a PhD that you wouldn't be able to do with only a bachelor's degree. And there's, you can continue in the more academic side of things by being an editor for a journal that will publish academic research papers, things like that. But I didn't really know, and so and I thought that you know, I thought that you know, it might be possible to also do some more research, get some more skills, and figure things out. So so one of the things that I ended up doing was called a postdoc, where you do several years of research after your PhD in the lab, in another academic lab. And I ended up going to the Max Planck Institute for Developmental Biology in Germany. And it's not really because I wanted to live in Europe or in Germany, or it was just kind of a coincidence that the professor that I was interested in working with happened to be there. This was somebody named Detlef Weigel. And in his lab, they were studying a whole number, all sorts of different things. But most importantly to me was that they were doing a lot of genome sequencing and analyzing sequences. And I didn't really have a lot of, even though I was working on DNA, I was working on more the molecular side. I didn't really have the computational skills to analyze sequence data. And that was a good place to go to learn those skills while doing my own research in those subjects. So so it was a really good experience for me. And then after a few years, I moved to Davis, and this was actually unrelated to my own career. I was moving for reasons of my husband's business at the time. But when we moved here, I ended up getting to know a number of people at UC Davis, including the professor who I'm working with now, Richard Mitchell-Moore. After talking with him, we sort of both realized that we had a lot of common research interests, and that it would be a really good fit to join his lab as a project scientist. So that is how I ended up where I am. And I guess if I can use this career journey as a way of dispensing any kind of advice, is just that it's okay to not know what you want to do. And that, and part of that though, is that you have to leave a lot of options open for yourself. So, so even if you're not sure what you want to do, you might want to prepare for several different possibilities, just so that those avenues are going to be available to you in the future. So I'll talk a little bit about what a typical day for me is like. So I would say that my days are pretty variable. So, so this is the building where my lab is. This is greenhouse facilities where I am now. And as part of the facilities, we also have fields available, research fields. And although what I'm doing now doesn't involve a lot of field trials, you know, at least a few times I can come and help, and I like being out in the field, so I go there. But mostly I will be either in the lab or in an office there, or in the greenhouse. And if I'm in the greenhouse, I might be checking my plants, seeing how they're growing, taking samples from them, seeing what kind of traits they're exhibiting, making measurements, that kind of thing. And if I'm in the lab, I might be grinding up seeds like I'm doing here, extracting DNA, and then using that DNA in other experiments. I might be looking at the microscope. So here are some images of pollen from lettuce. And then I also do a lot of work on the computer. So as I mentioned, if I'm looking at DNA sequences, I have to process them on the computer. This is a field known as bioinformatics. So a lot of times you'll be writing programs and using programs to analyze the data. I might also be using spreadsheets and plotting programs to collect data, to summarize it in plots. I might I have an electronic lab notebook, so I'll be entering in observations and notes from my lab experiments that day. And of course, when you've completed your research project and you want to communicate your findings to the rest of the scientific community, and you then put together all of your figures, and you put together your research in a way that summarizes your findings. What was the question you were asking? What experiments did you do? What were your conclusions? And what the context is in relation to the rest of the field? And so that gets put into an article that is then published in a journal. So, but you have to spend time writing it and putting it together. What else? I might be meeting with students. I might be meeting with other members of the lab. So we talk to each other a lot to not only share with each other what we've learned in our projects, but to discuss techniques. There's also a lot of seminars that we might attend, other researchers coming from different universities to tell us about their research findings. Usually before this would exist in the form of a journal article that you could read, the scientists will come and share their information with other scientists and with other people in the community, and it's good to do that. And then also I spend a lot of time on the computer reading papers so that I can figure out what other knowledge exists out there and how it might relate to my own research. So really, my days are pretty variable. So I would be doing any number of these activities on any one day. And that's what I kind of like about it. And it's also it's very flexible because it's up to you to decide how you're going to spend your time during the day. And I really like that about it. And the other things that I really like are that I get to do, I get to work as a team a lot. I get to work with students, usually bachelor's level students, teaching them new things. But also I work with other colleagues in my lab, including our greenhouse manager, who helps me out a lot. And also other scientists, PhD students, postdocs who are in the lab. So there's a lot of collaborative work, and then there's a lot of independent work, and I really like that combination. And I think that, so if you guys want to know more, either about plants and agriculture or science in general, I have some resources to recommend. So I checked, so these are some books that you can get from the Sonoma County Library. I really like this one. It's written by another professor here at UC Davis, Pam Ronald, and her husband, who is an organic farmer. And so I highly recommend it if you are interested in that. There is a new edition, so this is the first edition, and the second edition I think came out in 2018. So I don't know if Rosalie, or you can contact if she could help you find second edition. I did find the first, or if it's there and I just didn't find it, but I did find the first edition. And then also, I really like this book just about plants in general because they have so many interesting and unique structures, and I just highly recommend checking this one out. For video, I really like anything by David Attenborough, but this is a recent one on plants from last year. These are general science podcasts that I can recommend. And then also there was this article that I believe I sent your teacher about seeds that can live for a long time, if you want to look at that. And if you want to get in touch with me, if you have any questions after our Q&A is done, or if you want to learn more about what we're doing in the lab, this is actually a picture from last Halloween of members in the lab. We're kind of a big group, but you can read more there. So I think this is the end of my presentation portion. So we can go into the Q&A, and I'm happy to answer any questions that you guys have. Sorry, Milo, sorry you were on mute.
Sorry. We have some really great questions here. And Carrie asks, have you done any work in genetic engineering?
Yes. Okay. I had thought that this might be a question that you guys might have. So, so I have done work in genetic engineering, not for commercial purposes, but just for the course of my own research in the lab. So if we want to figure out what a particular gene does, a good way to do that is to make mutations in that gene, which is the form of genetic engineering.
And so it's something that you support?
I'm not sure what the questioner means by support. So definitely, so I think that, yeah, so is there maybe there can be a clarification?
Yeah, Carrie, if you could clarify that. And we have also a question from Izzy. Are there any other ways to change genetic material besides CRISPR and artificial selection?
Oh, yeah. So, so mutations occur naturally. So there is a lot of mutations that just occur due to replication errors or damage. There are things that mutation is something that is happening all the time. So you can take advantage of existing mutations in your population. So this is something that we, so this is, I guess, what you end up using for artificial selection, if that's what the question was. And CRISPR is a way of making directed mutations, but you can also make mutations by radiation, and this is a much less targeted way of doing it. So you end up with mutations all over the genome, and then it's a matter of investigation, figuring out which genes have the mutations that you're interested in. You can, there's other forms of mutagenesis as well. So, yeah, so definitely there are many ways of doing it.
Okay, Carrie clarifies, she's wondering, on a commercial scale, do you think genetic modification is something that we should use regularly?
Okay, so, so I guess I should stop and clarify. So there's a lot of terminology that can get used interchangeably. And so so in a way, everything, all of our modern food is a result of human engineering in some way or another. So we didn't get from this wild lettuce plant to this cultivated lettuce plant without any intervention. So, so through breeding and artificial selection, we have modified this plant's genome into this plant's genome. And the other ways of introducing mutations that I talked about before, like irradiation, is not something that's considered a genetically modified organism, even though it has mutations that have been introduced. What is considered a genetically modified organism now is if you have a transgene, so a gene that you've inserted into another organism's genome. And CRISPR is different because you can do it without inserting any foreign genes. You can do it with inserting foreign genes and then taking those genes out, and you can also do it without inserting any foreign genes. And so in that way is not considered a genetically modified organism. So these are all different ways of ending up with different traits in plants and traits that might be really beneficial to humans in terms of allowing plants to survive and keep providing us with food in the face of climate change. And I kind of view them as, you know, they're all different tools in the toolbox, and I don't think that we should categorically exclude any one of these tools. I think that, and even within any of these categories like of a tool, you know, there's many different ways in which you could use it. So, and the way that I think of these tools, whether it's CRISPR or inserting a different transgene, or through conventional breeding and artificial selection, these are all, you know, different tools, much like in different technologies, much like the internet. And you can use the internet for different things, but those things should be evaluated in terms of what might be beneficial or not so beneficial. And so I don't think that anybody would say, okay, well, sometimes people use the internet in ways that we don't like, so we should just get rid of the internet. And so for the same reason, I don't think that it would be a good idea to just say, okay, I don't like this technology, so I think that nobody should do it. I think that we should evaluate how it's being used and make our decisions based on that. So I'm saying this as my own opinion, not as a representative of the university or anybody else. And there are certainly experts who study this. And the book that I mentioned earlier, Tomorrow's Table, Pam Ronald, she's an expert in this. And I really encourage you to read more from that if this is something that you're interested in. Does that answer your question? I hope.
I think so. And actually, Dr. Pam Ronald is coming on November 4th to do a segment here for Mind of a Scientist. Cool. So you can ask her questions, and I hope you all will read the book. And Seraphim and Stefan are asking, can you tell us about any interesting discoveries that you may have made during your research projects?
Oh, yeah. Yeah, I think that, yeah, so one that comes to mind very recently has to do with, actually it's related to seed germination. And this is something that I wish I had, so I'm glad you asked this question because it's something that I wish I would have mentioned earlier, that seed germination timing is very important. So just as it's it's beneficial to retain the ability to germinate over long periods of time, you also want to make sure that germination occurs at a time that's going to be beneficial to the plant. So when there's enough water resources available, and so some seeds, for example, and this is not something that I discovered, but this is all background to what I'm going to explain. Some seeds actually require the seed coat on the outside to be ground up first before it can germinate. And some seeds actually require smoke from a fire. And this is because when there's a lot of disturbance that will end up causing a lot of the plants in the area to not survive, this might be a good time for other plants to start to grow. So plants have different strategies for timing and making sure that their seeds germinate at the right time. or if there's like enough water versus not enough water. So a lot of times you have a lot of things coming together to make sure that seeds don't germinate when they're not supposed to. And one of the situations when germination would be bad is if they germinate when the seeds are still on the mother plant. So, sometimes you guys might have seen this. I opened up a squash the other day and the seeds inside had already germinated, which was fine because I could still eat the squash. But if those plants had continued to grow inside, then it would have made it so that the squash was not so good to eat anymore. And one of the things that I discovered recently with one of my lettuce plants was a seed that germinated while it was still on the plant. And that was something that we hadn't really seen a lot of in lettuce before. So I don't know why that happened yet. So it's something that I'm interested in studying further.
Aaliyah asks, is there a process that is used when trying to make an old seed germinate years later?
Yeah, so a lot of times, when I was explaining about how seeds can time their germination so that they don't germinate when they're not supposed to, and that they do germinate when it's beneficial, this is regulated a lot by different plant hormones. So, if you want seeds to germinate, that you think otherwise might not germinate, you can provide them with hormones that will promote germination. You can also, water, of course, is necessary for any seeds to germinate, and you can provide some nutrients in the form of fertilizer. There are different ways of doing it, but those are the ones that come to mind at the moment.
Sophia is wondering how the lab greenhouse is different than a regular one. Does it have special features that control variables affecting the lettuce?
So our lab greenhouses, we have ways, I'd say it operates much like any other greenhouse. So you have some degree over the temperature control, but it's not very precise, but we can keep it cool enough so the lettuce plants don't get too hot. So you might know our summers in Davis, we have many days that are over 100 degrees. This is not really a suitable time. So if you want to grow plants in the summer outside, and if you want to grow lettuce plants outside in the summer in Davis, it doesn't really work so well. But in the greenhouse, you can keep it cool enough and just maintain the conditions that are right for growing lettuce. But other than that, it's not so super specific. We also have some growth chambers, kind of like a big incubator where you can, where there's some lights inside, and you can have more precise control over the temperature and the humidity. And this, these are all important variables for growing any kinds of plants. And then you just have to adjust these variables to promote and support the growth of lettuce.
Reichen is wondering, do you think it could be better to modify wild lettuce to be more palatable since it is already hardy rather than modifying domestic lettuce to be more resistant?
That is a very cool question. And yeah, I mean, I guess it's a different way of going about it. Yeah, it's kind of a more philosophical question, because at some point you have to decide at what point. I mean, it kind of goes down to a question of speciation, right? And how many? Yeah. So how to put this? So actually, the genomes of the wild lettuce and the domesticated lettuce are very similar to each other. And even though there's more variation in the wild lettuce, they are otherwise pretty similar. And oftentimes what gets categorized as a wild lettuce versus a domesticated lettuce is whether or not the seeds stay on the plant. So in domesticated lettuce, the seeds will stay there, whereas in wild lettuce, the seeds will get dispersed in the wind. And it turns out though that this trait is really a single gene. So this is why it's a kind of a hard question to answer, because it really kind of depends on how you define a species, and so this is one way in which the species has been defined, but there's much more genetic variation in this one. So I guess if you wanted to re-domesticate a wild lettuce, you could change this one gene to make the seeds stay on, and then work from there to get to the more palatable types. But this is it's something that I would actually like to think about and discuss more. So these are the things that have come to my mind now, but if you want to keep talking about it sometime, I'm happy to do it.
Aaliyah is wondering, how would you develop plants that are more resilient to drastic climate changes?
Yeah, that is a very important question, because you usually have many different sources of stress coming from climate change that you have to deal with at once. And most of the time for any one individual researcher, it's too much for any one person to do, because as I said, we have all these puzzle pieces, and it takes a long time to just figure out even one component. So how to make plants, tolerant to drought, is one question. But with climate change, you might have both drought and flooding. And so maybe you have some places where you would have to make plants that are capable of surviving both because of the changes in rainfall patterns. It's a very, very hard question, and really not one that I think that I could solve as an individual. But as a community, we're all working on our own little pieces, and then we come together to figure out how to put them together, how to make our puzzle pieces fit one another. And I think that the solutions are probably going to be different based on what species of plant it is, what are the most stressful conditions imposed by climate change for that species. And I think that there's not a one-size-fits-all solution. I think it kind of goes back to the question that was asked earlier about, genetic engineering and genetic modification. So we have a number of different ways of changing plant genomes and changing and introducing different traits into crop species. And we have to have a lot of these things available in order to keep growing plants that are resilient.
Okay, I think we have time for one more question. What do you think the most biologically interesting plant is?
Oh, I really like the Australian Christmas tree. So it actually will try to choke off the roots of its neighbors, and it has these little knife structures to cut. I just think that is something that you would never think of a plant as being capable of doing. So I think that is perhaps one of the most biologically interesting plants.
Oh, cool. Well, I think we're ready to briefly discuss the worksheet.
Okay. So let me share my screen again. So I think I'm going to get my escape from this. Yeah. Okay. So hang on, sorry, I have to change something. Okay. Sorry, it took me a while to get to the worksheet. So now we have our worksheet about the experiment for today, which has to do with seed germination and longevity. And the essential question is one of the two essential questions that I've been studying, which is about whether seeds germinate and what determines whether they do or not after long periods of storage. And there's a couple of background questions, so things that you should think about before starting. I know some of you have already started, some of you probably haven't started yet, but before you begin, it's good to think about these things. And then once you've started your seed germination experiment, what I want you to do is observe the stages of seed germination. So in general, you have a dry seed, which I think I can show you some of those too. After you provide it with water, the water will enter the seed, and seeds will usually become a little swollen, so that's an imbibed seed. And then after a while, the root will start to come out. This is the embryonic root, which we have a fancy botanical term called the radicle. And then the shoot, the green parts of the plant, the cotyledons, the embryonic leaves will come out, and which we call the plumule. So, so this is kind of a general scheme, and depending on what kind of seeds you have, it may be some variation of this, but this is the general idea. And so I want, so when you start the experiment, after one day, look at the seeds and see how many are in each of these different stages. And you can continue doing this for a number of days. So I think I left space on the sheet to do up to 5 days. You don't have to do all 5 days if you don't have time, but it would be good to do at least several days, because then you can see the seeds kind of going through this timeline. And in general, to simplify things, seeds that show the emergence of the radicle are considered to have germinated. Although, you know, there can be other definitions, but that's how I'm defining it for the purposes of this experiment. And so we'll have this more specific data about all the different stages for you guys, because I think it'll be really interesting to observe how the seeds go through the stages. But what I'm hoping to do is to collect information from everybody, because you've all had different seeds and different ages, and we can put them all together in a spreadsheet so that we can actually look at how these factors influence, so the age and the type of seed, whether they germinate or not. And that will be collected into a spreadsheet. And I will kind of go over that when we go into the breakout rooms. And then there's some questions about conclusions that you might draw from this experiment, and more thoughtful questions about why it's important and how you might extend it in the future. And that's basically it.